Reuben is going to be talking about cancer and some of the latest trends going on with that. He got his BS at Cornell, as you can read, his PhD in biology at MIT, and did his postdoc at Harvard before he came here to Salk. So we are very excited to have him as one of our speakers. And one of the things that I was surprised to see was not on your biography is the fact that as of January, he was named the director of our Cancer Institute here at the Salk. And this is something that's pretty special because we are one of the only places, I believe, in the world that has a cancer research center that's not associated directly with a hospital. So that's a really, really amazing thing to have the researchers that we have here at Salk working on that very important disease. So with no further ado, Reuben Shah.
Great. Thank you, Donna, and it's an honor to be here to speak to all of you. And on a personal note, besides being directly connected to Ellen and to Ron, and to be a happy part of the Salk family itself, within my own family, my father was a high school teacher. He taught history, and so I grew up in a house filled with leftover textbooks and other things, and so from a very early age was influenced in that way to be an educator myself. So we'll see how well that works out here. Okay, so what I'm going to try to do is to kind of give you a cross-section of both the latest in cancer research, but more to give an overview of what the world kind of knows about cancer and its treatment today, taking into account kind of everything that has been developed over the past 100 years, and then kind of where we're at in terms of our actual understanding of the underlying causes of this. And this slide, like many of Ron's slides that we just saw looking at diabetes, is actually just to remind everybody that cancer remains a leading cause of death in this country. And it's shockingly similar in terms of its rates to the number of deaths from heart disease. So these are the number one and number two causes of death in the United States across the board. And one interesting thing is that the incidence of heart disease has actually been slowly dropping from better testing and better control of the issues that lead to heart attacks, and effectively better medical interventions to keep people alive when you've had a modest heart attack. In contrast, in spite of decades of cutting-edge research in cancer and some great successes that I'll highlight along the way, and certain subsets of cancer have gone from being a death sentence to actually being something that is nearly curable, or at least able to be put into remission. Still, the vast majority of cancers we have not made enough headway on. We is the collective we, scientists in the US and scientists in the world. And to put it in perspective, there's this headline actually in the past 2 months, this is from the LA Times, in the state of California, as well as in all the other states that are shown here in dark blue, more people actually die from cancer than actually of heart disease or any other specific cause of death. So just the sheer numbers of people who die from cancer, that kills more people in this state than anything else. And now this speaks to something that Donna just referenced, which is the fact that many people don't know. Within the US, a large part of the research to get at the root causes of cancer and then to come up with new therapeutic treatments stems actually out of legislation. In a way, this goes back to Peter's introduction of his father and trying to think of ways to make long-term changes to a society. You have to have all different types of people involved and invested in different ways, passionate about their various causes. But it still is striking at the end of a 50-year stretch or something to look back and see how sometimes legislation really does change what happens in people's behavior and knowledge and what is created. No different than John F. Kennedy declaring that we're going to get to the moon. If that had never been declared or declared by anyone less than him, we probably would have never gotten there. And if we had not pushed so hard, the Russians would not have pushed so hard. And there's a domino effect of events in human history. Something that many people don't realize, or is certainly not the first thing they think of when they think of President Richard Nixon, is that towards the December in 1971, amidst everything else that was going on, he actually signed into law the National Cancer Act. And the headline that accompanied this is "Nixon declares war on cancer." It's what the newspapers ran that day. But one very concrete thing that came out of that piece of legislation was that money was set aside at the federal government level to designate in most of the major cities in the United States at that time so-called cancer centers. Now, most of those cancer centers, and those centers would receive funding for the purposes of patient treatment and cancer research, dedicated at institutions that already had a deep history themselves, not because there was federal money for it. They were already working on cancer and trying to cure cancer and treat patients already. So it was not given to create something that didn't already exist. It was given effectively to galvanize and reward places that were already dedicated to the cause. To this day in the U.S., just last weekend in Chicago, was a convention of the American Association of Cancer Centers. There are 70 dedicated cancer centers, federally funded. The NCI is the National Cancer Institute, and that is just the government body that hands out this money. For those of you who don't know, the federal government's federal dollars that go towards research in all diseases is under the umbrella of the National Institutes of Health. So the National Cancer Institute is one of 10 branches of the National Institutes of Health. And so there are 70 cancer centers in the United States. 63 of them see patients. And they are, of course, actual research hospitals that see patients and do treatment. And a subset of those also do research while they see the patients. There are only 7, actually, in the entire country that are dedicated centers, as Donna mentioned, for doing just research, for doing research at the cause of cancer and for developing new therapeutics to treat cancer. The list of them is actually here. MIT, actually where I did my PhD, was in the Cancer Center at MIT. Cold Spring Harbor in Long Island, some of you may know, is another one. And the Salk Institute was the third. Those were the 3 original designated basic cancer centers in the U.S., set aside almost at the time in the early 70s that the money was given out. Since that time, a handful of other places have received this designation, one of which is another research institute here, right on the mesa here in La Jolla, the Sanford Burnham, now Prebys Discovery Center. That actually makes the city of San Diego the only city in the United States that actually has 2 basic cancer centers. For those of you who don't know, we also have a federally designated clinical and research so-called comprehensive cancer center, the Moores, the UCSD Moores Cancer Center, which therefore makes San Diego the only city in the U.S. that also has 3 such institutions. So we have actually a collaborative consortium between the 3 cancer centers. And so this is something I myself didn't even know when I was recruited from the East Coast to come to the Salk to do this. But we actually are very fortunate to live in this city that has such both a history of groundbreaking cancer research, but also to this day, cutting-edge cancer research. Now, what's also kind of surprising to me even to think about is that there hasn't been anything at the national level in terms of changing priorities or re-stirring the pot of putting more money into cancer since 1971, which was the year that I was born actually, until this January, when in his final year of his terms, President Obama announced the so-called cancer moonshot, and to set aside federal funding dedicated to cancer to try to speed up cancer research efforts in the country. And I'm sure if you've heard of this, you would know that as part and parcel of that, he wisely put all of this in the hands of the Vice President Joe Biden. I'm sure many of you heard the stories last year. Vice President Biden's son, Beau, died of glioblastoma, which is, of course, the deadly form of brain cancer, within 2 years of being diagnosed with it. That is actually, to this day, the standard time course for people diagnosed with typical glioblastoma. One almost never lives more than 2 years. So Vice President Biden is extremely passionate, of course, and very personally vested in pushing this forward. And what's also interesting about the auditorium you're sitting in and being here at the Salk is that this was announced as part of the State of the Union address, the first public press conference held by Joe Biden to discuss what they were going to do, how they were going to take this mandate and do something with it. Interestingly, happened at the World Economic Forum in Davos, Switzerland, a couple weeks after the State of the Union address. And at that time, sitting to Vice President Biden's right is Francis Collins, who is the head of the National Institutes of Health, the organization I just told you that is the umbrella thing that funds all health research in the United States. Sitting to Vice President Biden's left is a woman named Elizabeth Blackburn, who is a Nobel Prize winner who did basic research that led to the discovery of structures in our chromosomes that relate to cancer and other diseases. Elizabeth Blackburn also is the president of the Salk Institute, right here. So our president of this place, who walks around this building all the time, in fact is there, seated right next to the vice president on the occasion to announce to the world that the United States is going to take on and defeat cancer faster. So it is an honor for us. I mean, this is just further proof, in a sense, to me, of the legacy of Jonas Salk and the legacy of famous people and continuing that fight straight into 2016. Oh, and one of the great things, this is actually all available on YouTube. If you actually just type in Biden, cancer moonshot, Davos, you'll get like the whole 50-minute video of them talking through it. I don't encourage it unless you're, have time to kill or you're drinking and you can fast forward it. But at some point, he goes around and has 20 leaders from different cancer centers in the United States all weigh in. And when it was Dr. Blackburn's turn, what she noted to him is that she made two different analogies. One is that you cannot come up with effective cancer treatments without doing basic research that underpin these things. It's two hands shaking and two hands clapping. The other is that you cannot also search for cures for cancer without developing proper cancer prevention methods. And so those are her two most popular analogies, and that will certainly guide some of our research. So a little bit about cancer, a little bit of background. I'm going to try not to delve right into the heart of the molecular details. I'll start with this background, which will seem simplistic. But I think all of you probably realize the cause of cancer at this point is quite well established, and that is due to alterations in specific genes in our body. Outside things that we are exposed to, which include viruses, which include chemicals, including sunlight itself, obviously, can lead to changes in individual base pairs within the DNA of individual cells of our body. Changes like that occur all day, all the time. They're happening in our bodies right this second as we're sitting here. And the cell has machinery that goes around and actually recognizes what the proper sequence is supposed to be, and everything gets changed. The cell's like, ha-ha, we got a problem. Cell goes around and flips it back and repairs the DNA. That repair machinery has a certain statistical success rate, which is extremely high, or else none of us would live past the age of 6 years old because we'd be riddled with cancer. And in fact, there are children who are born with defects in that repair machinery, and sadly, those children are some of the types of childhood cancers that afflict children that develop widespread tumors. So we actually have evidence of what happens if we didn't have that type of machinery. Still, at some, again, statistical frequency, exposure to sun and exposure to other chemicals and exposure to viruses will result in different types of cancer in individual cells of our body over time. The other thing is that we inherit. Heredity also contributes. So not just in major ways. All of us may know someone whose family, it seemed like half the members of the family had some type of cancer or another at a young age, before the age of 40 or before the age of 50. And it seemed odd, but it was tracking with roughly half the people in that particular family. And then when they looked back, it was also true of their cousins and their grandparents. And those are clearer cases of a specific gene causing that sensitivity to cancer. But more often, you'll have subtle risks that you're not certain that you're going to develop breast cancer, but your risks just went up 20%. And this is much more the more difficult reality of our ability now scientifically to deduce specific numbers and specific statistical incidence rates for different types of cancer. And the big question, which I myself struggle with, is what exactly do we do with that information? If a doctor or a scientist could tell you with absolute certainty that your risk of colon cancer is 15% higher than the average, where's the cutoff? Certainly knowing that you have that increased risk might lead you to change your diet a little bit, but how much you then live in fear of the cancer or not, of course, is something that is a human experiment going on right now in the world. Now, cancer itself, to just, again, stay with the general overview of concepts, the easiest way to think about what goes wrong in a cancer is actually just to think about basal cell carcinomas that most people get. Actually, most people, if you live to be 100, your odds of getting a small skin lesion that is actually precancerous is almost 100%. So if you do like a full autopsy on someone who dies of natural causes, like not cancer, at the age of 100, your odds of having small pre-neoplastic lesions someplace in your skin is exceedingly high. Another thing many people don't realize is if you live to be 100 and you die of natural causes, you die in your sleep of heart arrhythmia or something when you're 100, if they do a full autopsy, your odds of having small polyps in your GI tract is also almost 100%. So all of our bodies, every human effectively, has certain sensitivities that predispose us to overgrowth of certain cells. Now, what's in common between the surface of our skin and the lining of our GI tract? Why are those some of the weak links? Well, those just happen to be some of the cell types that are the most exposed to the outside environment. Your GI tract is exposed to everything you eat and you put into your body this way, every chemical, everything. And your skin, obviously, exposed to everything on the outside. Not coincidentally, those two cell types in an adult, when we're not growing anymore, those are some of the only cell types that are actually still rapidly dividing and turning over. So our bodies also evolved. Our bodies, our dogs' bodies, every higher species, it's the same thing. The cell types that naturally get shed, whether you realize it or not, the lining of your intestine turns over almost once a day. The individual cells only live for like a day or two, and then they naturally turn over and they're replaced by a new one. So those cells are normally dividing. Same thing is true of our skin. The cells of our skin are actually only programmed in the first place to live for a day or two, and that is specifically to prevent you from being riddled with cancers because the cells are turning over anyways. So these processes can go slightly awry, and that's, of course, what allows for cancer cells to evolve in the first place. But it's important for us to kind of put this in the context of how these things occur naturally. So in the case of the surface of your skin, we're exposed to UV radiation. If you went around your entire life in San Diego, never wore sunscreen once, it's just a statistical game as to what the odds are that you would get a single cell that had a change from radiation from the sun in the wrong spot. Our DNA contains about 25,000 genes. There's far more regions of the DNA, though, that don't actually have a gene in them. They're just long interspersed regions that don't have genes and no known function that scientists have figured out yet. So every time we're exposed to the sun, the odds of it actually landing inside a gene is small to begin with, less than 1 in 100. The odds of it landing in like an important place in an important gene, It's far less than that. It's more like 1 in a million. That's within one cell, but you have many cells on the surface of your skin. So again, you can easily see how this is just a statistical probability then of developing skin per year, per sun exposure, per unit time. Even when you do have a DNA change, as I was just saying to begin with, you have this machinery in your cells that goes along and recognizes something's wrong and it fixes it. So it's not so much the incidence of having the change in the first place, it's, again, the probability of that machinery failing. So we have all these safeguards that really allow us to go for extended time periods with incidence-free. Even when all those systems fail, what happens is one cell simply starts dividing and becomes 2 cells. And it doesn't start dividing out of control. It's not growing like a weed the first day this happens. It's barely growing at all. It turns into 2 cells, and maybe over the course of a year, it turns into 50 cells. And this may have happened in most of the people in this room's bodies. We would never know this. You would have no idea. 50 cells doesn't look like anything. It's not until some years later, if you're unfortunate enough, you get another mutation in one of those other cells, that's when things really start to take off. And they grow locally, and you call that's when the term neoplasia starts coming in. And it isn't until usually some years later, in most types of cancers, that you actually get 3rd, 4th, and 5th events occurring just in a subset of the cells within what we would call a tumor, that things get quite bad. And that process is when it becomes invasive. The term is called metastasis, and the cells start spreading throughout our body. And that is the most dangerous and deadly aspect of cancer. Now, just some general terms. We know that there are effectively a limited number of genes. Of that 25,000, there are less than 500 genes that you would say the main function they do is to regulate the growth and survival of cells, and if you get a mutation in them in the wrong way, can give rise to cancer. So that's an important number. So there are less than 500 cancer-related genes, and they fall into just 2 groups. There are genes that when they are altered in the surface of your skin or in any other tissue, that they are turned off. So that the loss of their normal function is what causes the cancer, and therefore they're given the name tumor suppressors because their normal function is actually, believe it or not, we have a whole bunch of genes in our body that exist to stop cancer from growing, or if we get one of those bad DNA changes, they're actually little sensors floating around the cell to make sure everything is just right. And if anything's slightly out of whack, they set off a little cellular alarm, send a signal to machinery to be like, got a problem. Problem in sector 8. There's a little aberration in the DNA. There's an aberration in the mitochondria. Send in the repair machinery to fix it. So when you get a mutation in one of those sensors, that is the easiest way to give rise to a cancer. The other type is the bad roll of the dice. If you actually get an alteration in something that normally, when we're growing from small kids up to big kids, tells the cells of your body to grow, if you get so unlucky that the sunlight happens to zap one of those growth regulatory genes and actually tells it, oh, no, I know you're not 5 years old anymore and you're not supposed to be growing anymore, and there's all these other signals telling you not to grow. Ignore those signals. Pretend your body is 3 years old again. Start growing. And that is another major source of cancer. Those pro-growth, pro-cancer genes are called oncogenes. So that's it. There's really just genes turned on and genes turned off. So this is all depending on your perspective. This is the good news, or this is the bad news, is that we can layer the roughly 350 to 500 cancer genes. We know enough about what their functions are and their maps. We can make this complex map of how they interact with each other, what the regulatory events are, how they pass the baton to each other to execute different things within the cell. And even amongst scientists, when I put this slide up, I've been trained in this kind of a background, of course, for many years now at this point. So I think this is exciting because it's actually not that complicated. And I'm like, this is great. And most people are like, God, I hate this kind of work. This is just like, why does it have to be this complicated? Because it seems like it is. But the analogy I often make, even with young students who come to my lab, I mean, young graduate students or undergraduates that come to work in the lab for a while, they see something like this, and then they think, is there a different kind of lab I could work in? So maybe like muscular dystrophy is just like one gene, right? Can I just work on that one thing? But I make this analogy when I was like a second-year assistant professor here at Salk. There was a meeting, a cancer meeting that was in Tokyo, Japan, and I mean I was really naive, had no idea about any of this stuff, and I also was like, oh, I'm a professor now, and I'm sure I get there and someone meets you with a little sign and they'll take us wherever we need to go. And I went with two PhD students from my lab. When we went there and we landed, we realized, no. There's nobody with a sign. We're completely sleep deprived. We know the hotel we're staying in is someplace in downtown Tokyo, and we couldn't find anybody right then. It was like an odd time that we got in who spoke English. And I started getting panicky while trying to seem cool to the students. It's like, oh, I'm sure we'll figure this out. You guys don't happen to speak Japanese, right? No. I'm like, well, with the airport, we landed out here, and I was like, well, if you think about it, I lived in Boston for years. And my in-laws live in New York, and I was like, well, it's really not like any other major city's subway system. There's these outside hubs, and they all kind of, one way or another, get into the heart of the city, and that's really the downtown. And in fact, it's the same thing with the cell and these cancer pathways. There's all different things that are coming in from the suburbs and from outside, but they're all going into the downtown. The downtown is the nucleus of the cell, where the DNA is kept, and where all the major decisions come down. They all get, metered out in the nucleus. And so that really is, you have avenues and major avenues that get you into different parts of downtown. And so within cancer therapy, we need to understand which major thoroughfares are where the problems went wrong, and then we need to set up blockades to block those routes downtown. And of course, the cancer cell is going to mutate and try to get around that, and so we have to find the backup ways to come up with the right combinations to stop the cells from getting to divide. Another thing, though, that scientists have done is, besides decoding the biochemistry and the individual details of how the genes connect, is to know that you can group these things. Oh.
Use that one.
Okay. Good. Boom. We can group the cancer genes based on what they do. And this very influential article was written about 15 years ago now called The Hallmarks of Cancer. This is one of these things that is really held up over time. This is one of the central things for me to tell you today, is that instead of learning the names of all those 350 genes, you can group all cancer genes based on what their normal function is. So some of them function to tell cells when is the right time to divide. Other ones tell cells, what in God's name are you doing? You are an adult brain cell. You are not supposed to be dividing under any signal. And the signal coming out of the gene is like, no, I'm getting, it's supposed to be time to divide. And there are actually more genes whose function is to be like, sit down. It's not time to divide. And those are these insensitivity genes. There are other genes whose function is to tell the cell how many divisions it actually is allowed to have total. There's machinery for that. There's also machinery for telling a cell. There's a process in the cell called cell death. The technical term is apoptosis. Your cell has these sensors that sense all different features. When a certain number of them line up, and it is, the simplest way to think about it is this way. If the cell gets like 4 different signals that tell it that is completely out of whack, it actually will kick in a process to kill itself because things are too far out of whack. And that process is called apoptosis. And all advanced cancer cells have to basically bypass that machinery, because just in order to get that advanced as a cancer cell, you will have tripped enough of these sensors and alarms within the cell that it would naturally try to kill itself. And so you basically have to take, the knife away from the cancer cell so that it doesn't kill itself. And so putting the knife back into the hand of the cancer cell is a whole different avenue. And then, of course, the last stage of cancer, which is the most deadly, is that the cells start moving out and get all over the body. Now, it turns out that that influential article from 2000 got updated just a couple of years ago in 2001 when the authors realized that they had not really captured everything that was going on, and so they actually added 4 more processes with greater understandings. One of them relates to cell metabolism that I'll talk about in a minute. Another one relates to inflammation and the realization that inflammation in your body can actually lead to cancer. And the final one is a greater appreciation, only in the past 10 years, of how important the immune system is and how much it actually normally plays a role of preventing our bodies from having cancers. Even after the cell control mechanisms have been lost, our immune system still plays a role in actually getting rid of tumor cells all the time. I'm just going to skip ahead, actually, to kind of some of the real more take-home points then that I think is the exciting breakthroughs that are more recently. And one of them has been in the past 10 years only. People always talk about there's this breakthrough or that breakthrough. I'm trying to give you what I think the big picture, real facts on the ground breakthroughs have been. One of them has actually simply been technological and the ability to sequence people's genomes. I'm sure many of you have heard about this, especially being in San Diego. What you might not realize is actually it's a company here in San Diego known as Illumina that was the ones in response to a national challenge to create new instrumentation to be able to sequence a human genome for less than $1,000. To put this in perspective, 15 years ago, it cost almost $1 million. It didn't cost like $10,000. It didn't cost $100,000. It cost almost $1 million. So to make it reproducible and fast and cheap was like a really high barrier. But this company is one of the pioneers in a technology known as next generation. They called it sequencing, DNA sequencing. And with those advances in these massive machines, probably bigger than that piano, you can now get the full sequence of any person's DNA within a day. And that is really remarkable. And again, that's something that most people don't even realize occurred right here in this city. With that, patients can come in. You can go, if you go to Moore's, depending on most types of cancer, they will take a sample of your blood. They're taking, in part, that sample of your blood also to get the sequence of normal cells out of your body. They can screen the blood for tumor cells that are circulating, but they can also get your normal DNA sequence. And then they can compare that to a sample from the biopsy of the tumor. And they actually will scan the whole thing. They will focus in on those top 300 cancer genes, or even a subset of those cancer genes, and then ask, does this person's tumor have these common mutations that are found in these different types of cancer? Because if they do, then we can try to treat the patients based on the types of specific gene mutations that are found in their tumors. This is probably the only other thing for me to really impart upon you today. This concept of trying to treat a patient based on the specific genetic changes in their cancer is called precision medicine. It is often also called personalized medicine. Personalized medicine doesn't just have to do with cancer, it has to do with all things. They're the same. They're one and the same. Those terms, if someone's like, no, I think it's different. No, it's not. These are the same things, and it's literally just using the sequence of changes in our genes that occur in the diseased tissue as compared to your normal body cells to then help guide treatment. There are old examples of this that people just didn't use those terms, including first for breast cancer, so-called ER-positive breast cancers. If you had breast cancer and you found out it was ER-positive, this was a good thing. I mean, this is about 15 years ago now, some of these advances. And the reason it was a good thing was because it was one of the only types of breast cancer that had a specific treatment at all. It had a drug named tamoxifen. Interestingly, the estrogen receptor that we're talking about here is a nuclear receptor that is that entire gene class that Dr. Evans discovered and has worked on for the past 30 years. So these things not only control our ability to exercise and our ability to gain weight and our ability to do all kinds of things, they also govern the risk of different types of cancers. And this is a very direct example where if you have this gene or not, you respond to this therapy. Because, as you can imagine, the drug acts on the protein from this gene, the estrogen receptor. You say it's ER positive because it expresses it. If it does not express the estrogen receptor, you can still take the drug. The drug doesn't hurt you. The tumor cells don't have the gene to act on, so it's just going to do nothing. You're effectively taking a placebo. Another example that came up in the early days was this so-called HER2-positive breast cancers, and that's a cell surface protein that they made a therapeutic, an antibody against, that could attack that subset of cells. So again, if you have this protein on the surface of your breast cancers, the drug will have efficacy. If you don't have that protein on your breast cancer cells, the drug doesn't hurt you. It just gets in and does nothing. Simply does nothing. So you can see why it's so important to be able to know whether you have these specific genetic changes. And then the third group is a subset of leukemias. CML, chronic myelogenous leukemias, have a particular changing of two genes being fused together, and a drug called Gleevec is a specific targeted drug against that subset of tumors. And interestingly, the foundation for making that type of a drug grew out of research from another famous researcher here at Salk named Tony Hunter, from whom actually I only this year took over as being the director of the Cancer Center. Tony discovered a modification of proteins known as tyrosine phosphorylation. It's just a particular chemical group. He discovered it. There was no precedent. It was a crazy result at the time. Most people would have simply ignored it and been like, is this no? This doesn't make any sense, and just ignored it. He did not ignore it. He pursued it for years, and then that changed a whole new massive chapters in the history of cancer research, which led to the development of drugs like this, which are now nearly curative for subsets of cancer. CML used to be a death sentence within a handful of years. Not only is, are there generalizations of statistical trends, there are people at Salk, and there are people on the board of trustees of Salk who were diagnosed with CML nearly a decade ago. They take Gleevec. They are in full remission, and as long as they remain on the drug, they have lived full, healthy lives since then, which would have just literally been impossible. It is not possible that they could have lived more than 5 years prior to this. And that's a sad thing, is the people who come down with cancers the year before the near cure is discovered. So that's what we need to do as a community of scientists, is to come up with a whole bunch more of these. This is great. We got a handful of examples that were great, but we need to come up with more of these. So today, if you go into the clinic with broad cancers like colon cancer or breast or lung that are so prevalent in the population, prostate cancer, you will get a piece of your tumor sequenced almost certainly, and that will split you into different groups. In the old days, you would just get chemotherapy, which would have a benefit for a fraction of the patients. It would do nothing for more than half the patients, and for some of the patients, it would actually make them worse. And doctors had no idea why. There was just a disconnect. You had stage 3 colon cancer. It's called stage 3 because of how big the colon cancer was and if it had invaded near the muscle. But the genetic basis for that colon cancer, or what you could do about it, complete black box. Thankfully, today, we know more, and we can split patients based on what the genetic changes are. The big challenge now is that even with this knowledge for different subsets of colon cancer, for example, we have very few targeted therapeutics. So we can tell you all the details of how you probably got that particular subtype of colon cancer and what's going to happen next within the individual cells. But depressingly, we don't actually have the magic bullet to treat those subsets of colon cancer today. So that's the future, is to be able to read the sequence of these genes and come up with more of these magic bullets to treat cancer. So the good news is there's a big pipeline at a lot of different companies, and many different therapeutic things are there, but the trials, as you can imagine, have to be done. They have to be done in a safe, predictive way. You have to have proper placebo controls, and that takes years. So by the time a potential therapeutic for cancer is discovered until the day it actually is being widely used in patients is at least a minimum of 5 years, usually more than 10 years. So another thing that happens is you can develop resistance to the therapies, and I would say another big breakthrough from the past 10 years is that we now know the common ways that most therapeutic resistance to most types of drugs evolves. So in the old days, clinicians and scientists would just, new drugs would roll out, they'd put it in the patients, everybody would be like, this is amazing. Patients are living like, they used to die within 6 months, now they're living for 3 years, and then everyone would wait to see what happened, then they're like, oh, no, resistance is coming up, and now we have to come up with a new drug to treat the resistance. Today, anytime there's a new drug developed, from time zero, we know, stop fooling yourself. Of course the cancer cell is going to try to find a way around it, so you should start making the backup drug at the exact same time you start building the first drug in the first place. So that has been built into many of the major pharmas now, only in the past 5 years, say. And so there's greater hope for not having that exciting first newspaper headline, scientific frontline treatment for this subset of leukemia, and then another headline 9 months later, the bad news is it only works for about 9 months. Now, from the very beginning, the drugs will be tested together. And I don't need to go through all that. The last thing I just want to mention, because many of you may have heard about it, is that one of this is my own personal list of kind of the exciting areas of new methods that have never really been used therapeutically up until the past couple of years. The only one that is in widespread use and is widely being investigated is the only one in red here, cancer immunotherapy. These other three, cancer metabolism, CRISPR technology, we'll hear about from the next speaker, and oncolytic viruses is another brand new cutting-edge technology. All of them are different approaches, therapeutic approaches, to selectively killing different subsets of cancers. So cancer immunotherapy has been a very popular one due to great successes within subsets it's going to be most useful in subsets of tumors. And it appears, just to give you the punchline, it appears that it's going to be most successful within tumors where the patients have a high number of changes in their DNA. So skin melanomas and tumors that have wide mutational changes will be the most impacted by these. The last thing I'll say, though, is that my own lab's research, which I don't have time to get into now, relates actually to the second one, cancer metabolism, in particular subsets of lung cancer and other types of cancer that show changes in their metabolism. And for those of you who may have heard of this book, the gene that my lab has worked on for many years actually is an initiating gene in different types of cervical cancer and lung cancer. And in fact, Henrietta Lacks, who's the woman whose cells were actually used to make the first cancer cell line that was widely used in the United States and in the world, even though she and her family didn't even understand that. She died of her cancer before she even knew what they were going to do with this. This is a great book, so I would encourage you to read that. But that actually was due to changes in this particular gene, which my claim to fame and why I got hired here at the Salk was that I discovered what it did, which was heretical at the time, and that it was a bona fide tumor suppressor cancer gene, and what it was doing was regulating metabolism. And what's more, the activity that it possessed had been searched for years because it was the activity that explained how this diabetes drug that Dr. Evans spoke about a minute ago regulated diabetes. So I had discovered this unintended direct link between cancer and diabetes, which suggested that maybe patients taking metformin might have lower cancer risks, which it turns out they do in certain subsets of cancer. Metformin doesn't get into most tissues, so it doesn't generally stop all rates of cancer. But this was really so heretical that there would be such a direct connection between cancer metabolism that it took many years to kind of corroborate that. It turns out, though, the ideas that cancer might have relationships with metabolism goes all the way back to a German scientist almost 100 years ago now, Otto Warburg. He won the Nobel Prize back in the 1930s for some of this pioneering work. And long before people even knew about genes or DNA, they knew about this. And it was really clinicians that knew that tumors took up glucose that rediscovered the importance of this effect. So what I'm going to end you with is that we now know that there are many ways to treat these different subtypes of cancer with metabolism drugs. And so in my own lab, we have used derivatives of metformin, more potent versions of metformin, to try to treat subsets of cancer that lack that particular anti-cancer gene. But also in Dr. Evans' lab here in the past year, his lab, even though they mostly do metabolic disease and exercise and diabetes, they also do pancreatic cancer. And another major breakthrough from here in the Salk in the past 12 months has been the use of another one of these drugs, in fact, an analog of vitamin D for the treatment in pancreatic cancer, and this has now gone into the clinics. So this is another thing you wouldn't expect. In fact, you just heard him talk, and you would never expect that the person that you just heard talk about the exercise pill drug, in the other fraction of his brain, has also literally worked out a new potential cure for pancreatic cancer that's not just an idea, and it's not just something that might happen in 10 years. It's actually going into clinical trials and is in early stage clinical trials right now. So it's exciting to be here at the Salk, and one of the analogies is this. So let me just stop and say the grand hope for the future, to spread the word to future kids, is that there's a bunch of people working on ways to treat cancer, and we in many places now believe it will take a combination. You'll take one anti-metabolism pill, and you'll combine that with one of these like HER2 or tamoxifen type pills, and you'll combine that with other types of techniques, including conventional chemotherapies, which work really great on certain subsets of cancer. So we believe this is really the future. So let me just stop and say, we'll have a break, but the intro for the next speaker is that he will talk to you about one of these brand new cutting edge technologies, because I can say it for him instead of him saying it about himself. He was actually in the laboratory at MIT that discovered the technologies and was one of the pioneers who actually brought this to the world. And so we are so lucky. In the same way that somebody plucked me up and plucked Ron up many years ago, we have brought this young man here to Salk to help continue to be at the cutting edge for the world. Thank you.
And we can have a few questions for Reuben. There we go. I was like, if you leave the mic with me, I've got questions for him too, but I'll let you guys. There we go.
Hello, thank you very much for all this information. It was amazing. I'm just wondering, all these things that you're listing are fantastic to hear that they're coming. How much collaboration occurs between all of the other centers as far as cancer research?
Yep, that's a great question. And that's actually a major part even of the new Cancer Moonshot initiative is to further enhance that. I think one of the best parts about being at a basic center where we don't have patients, and also because Salk is so small, it really is this building and the famous marble courtyard and the building that's attached to it, that we know we can't do everything. We only have 50 professors. There's more than 50 types of cancer. If we even had one professor dedicated to each type, we wouldn't even have full coverage. So we always collaborate. We have to in order to exist. Also, the clinical trials, like with vitamin D analogs, we can't do those here. We don't have a hospital here. So everything is a collaboration. And so I think that's also been part of the secret sauce, honestly, in my opinion, of Salk, but also of San Diego, is that because we're not as entrenched as many of the East Coast cities that have had rivalries, not just with the other cities, there's rivalries of the two major hospital systems within a city, and they're like, no, I'll be damned if we're going to give the secret away to Sloan Kettering of, well, we got the new immunotherapy. They can read about it someday. Like, we're not going to tell them. San Diego has been newer. We've never had those types of historical things, and so I think we all play very nice with each other, the research institutes, because we think it brings greater things both to San Diego and to the world. And so we just don't have those types of historical. I guess that's the upside of being a new city, is that we're free to just work with anybody and there's no guilt or penalty.
We got one over here.
I had always assumed cancer was a type of affliction, but I'm hearing you say certain subsets of cancer constantly. So I always assumed there would be a cure for cancer someday, but are you saying that cancers are too varied, that it's never going to be one thing?
Yeah. I hate to be that definitive in any statement I would make, but the odds of there being a cure that can broadly affect most types of cancer is close to zero. cancer really is a collection of 300 diseases that we call the same thing. The odds of that happening would basically be the odds of if we called all the different bacteria and viruses and all the different infections you got, if there was one drug that could wipe out every bacteria and every type of virus, that would be the same what you would have to be hoping for if you were hoping for one type of drug to kill all cancers. And I think it's one of these things because up until the past 50 years in history, we called all these different diseases by the same word, just by one word, cancer. And then in the early days of some quackery and some honest people trying to say, like, we're going to cure it. They either didn't know or they were basically deliberately selling an idea that was either naive or dangerous to make most people believe there could be such a thing. That's unfortunately not likely at all. But there are still ways to treat all, in theory, there are ways to treat all fast-growing cancers, so you could wipe out 30% of them or something. But the idea that there could be a magic cure that could actually broadly treat all different types, that's probably not possible.
I just want to piggyback on that. So, okay, the idea of a magic cure, yeah, typically zero, right?
Yeah.
But then what about the idea of a widespread prevention? For instance, if you don't smoke, right, your odds of getting cancer improve dramatically.
That's right. And not just lung cancer, I'll add. Many people don't realize that. Obviously, your lung cancer has just dropped broadly, but smoking actually causes damage in other peripheral systems, which actually lead to the second most cause of other types of cancers is actually smoking. You wouldn't realize that. You're like, what are you talking about? Like, how on earth is the second most causative factor for endometrial cancer, whether you smoked or not? But other cells are being exposed to the carcinogens, whether you realize it or not.
So that plus, as I understand it, and I want to know much more about this, but there's also a pronounced effect from the current iteration of the Western diet. So say if
That's
you're
right.
vegan, right?
Yep.
Your chances of getting cancer are also dramatically lower. There was a
That's
study
true.
done in China that I think speaks to that effect, right?
No, that's true. And also of particular types of cancer. So colon cancer, for example, is highly associated with meat, basically. Certain forms of, often from the cooking, actually carcinogens from us actually, cooking things on the grill. But there are also other carcinogens in all kinds of other foods, including many vegan foods. So it's not quite that simple that you could solve all of that, but you could cut your risk of certain types of cancer enormously. But what might happen then is that the average population would live longer. Your incidence of lung cancer would drop literally so that it was buried way down on the list. Colon cancer would drop, it's buried way down the list. But then most people that live to be 80 would start getting what we now consider weirdo leukemias, because in the past those people all passed away from lung or colon cancer. And they just lived longer to now discover that actually humans have a susceptibility for some other thing because we'd never had that many people live that long to find out that weak link. But in general, I agree with you in terms of thinking of a broad-acting treatment, prevention. And to actually piggyback on the last talk we heard, many people don't realize this. Not only is diet a negative thing, exercise. Exercise is one of the most, it is like a magic pill in the sense that for reasons scientists, frankly, do not understand. It has benefits that lower risk of all different types of cancer. So there's hormonal effects, there's metabolism effects, but there are broad-acting effects. So a combination of diet and exercise may be the simple best thing until a magic pill is discovered that one can do that will lower your risk across the board. And a great thing to tell anyone you know who has cancer, like actually amongst people who have certain chunks, large swaths of cancer, exercise is one of the greatest predictors of delaying the time of keeping things in remission. In other words, exercise can help keep many different types of cancer in remission longer. Staying active, staying lively. I mean, part of this may even be psychological, but just engaging with the world, believe it or not, can actually change the outcome.
All right, I think we have time for maybe one or two more questions. We had some people with microphones back here.
When you were talking about Lumina, you were talking about how genes, like specific genes for one person can be identified, does that mean that we can detect cancer earlier? Like if we constantly do the gene identification?
No, that's a great question. Which technology-wise, we don't have the greatest way to do that. I mean, one way might be to sample the blood. There are people pushing for that. Like, if you could sample the blood just as a snapshot of what you've been exposed to tell. There's no way to go in to check every cell of your body. I mean, there's literally millions or billions of cells, and that's the bad part is it just has to go wrong in like one. So like one colon cancer cell, you could even pluck out a little piece of your colon, check the sequence of all those cells. It might be just fine. But then, like, just a little bit over from where the needle went in to get the little few cells, another cell was wrong. So it's almost impossible to check every cell that might have an alteration. But if you took a broad sampling of normal cells from the blood and just asked how much there were changes that might give rise to things, that's also a way to check if you've had cancer, sampling the blood, something called liquid biopsy. They actually check to see if you have circulating cells. Tumors actually shed individual cells that go out into the bloodstream. Many, not just the bad advanced ones, even the smaller ones will often shed 1 or 2 cells. And scientists have come up with different engineering things to catch those tumor cells, like effectively, honestly, with like a magnet. And then you can actually check and see how many of them you have and what the sequences are and if they've evolved, if they've mutated further, and therefore increase your odds of the cancer coming back.
Rachel, last one.
I was going to piggyback on that. Do you foresee a time with personalized medicine where you basically will go for certain checkups where you have this blood test and you look to see what's going on in there and you prophylactically treat people, whether or not it's really clear?
That's a really good question. I mean, I think that is like kind of a very 50-50 future possible, like even 10 years from now. And certainly 20 years from now, as part of, once you turn 40, like, for men now, you start getting a prostate exam or something, like manually. But, sorry, I'm over 40. It's too personal. It's too personal. But it may well be that once you're, 30 even, that they take, it's part of collecting your blood and looking for other aberrations, seeing if you're predisposed for diabetes, et cetera, that they actually run a sequence and check these things and look at these statistical odds of things. And if you do have something, they're like, you know what? You have this particular 10% extra susceptibility for colon cancer. You really need to cut down on the juicy steaks twice a week. Like, seriously, you got to change your diet. You got to change certain factors. So I do think that is possible, maybe even probable. And then along with the ability to diagnose, if you ever are so unlucky to come in with even a little nodule of what looks like a tumor, we have to, that's the one thing you can bet, is that the scientists now, it's like now that we've realized the size of the enemy, the size of the scope of what we need for personalized medicine, I think people are more impassionate, like we're going to get all the other types of guns we need to take out these other types. We have too few good drugs against the different subsets of cancer. We have to seal that off in the next 10 or 20 years. We just have to have more drugs that work against the different genetic subsets. Thank you.
Thank you.